Hello friends, in this video tutorial we will discuss about passive transport across plasma membrane. What is a passive transport? Passive transport is the movement of molecule from the area of higher concentration to the area of lower concentration along the concentration gradient. And this process is energy independent process, and in this process transporters are involved for the charged ion. Passive transport is of two types: simple diffusion and facilitated diffusion. Simple diffusion, it involves the movement of hydrophobic molecules across the plasma membrane, and they are non-saturable in nature because they do not involve any transporters. No transporter is involved in simple diffusion, and there is no saturation point. As there is no transporter, so there is no saturation point in this process, and the speed of molecule is high, as you can see in the diagram. The second one is facilitated diffusion. Plasma membrane proteins involved. In this process, the plasma membrane proteins are involved, and it allows the polar and large size molecules such as carbohydrate, amino acids, nucleosides and ions to cross the plasma membrane. And it allows both hydrophobic as well as hydrophilic molecules across the plasma membrane. And transporters are transporters are involved in this process, and saturation points occur. Facilitated diffusion is also of two types. First of all is channel protein mediated transport, and second one is carrier protein mediated transport. Carrier mediated transport, it is present in the free ribosomes in the cytoplasm. A carrier is a protein interacts with its target solute in a stereospecific manner, means it interacts with its target solution before the binding. The carrier protein interacts with its target solute before binding. And the moment of the solute in this process is very slow. The solute movement are bound at the one end and release the molecule to the another end. The binding of the solute induce conformational changes in the carrier, and it is responsible for the movement of solute along the concentration gradient. Carrier are saturable in nature, and that means if all the carriers are bounding to the solute, then solute concentration increase, which does not increase the movement of the solute. The example of the carrier mediated transport are GLUT for transporting glucose and fructose aquaporins. The next one is channel mediated transport. This process is extremely rapid, and the solute or molecule diffuses through the pore. See, this is a pore, and molecule or solutes, these diffuses through this pore. And the channel mediated transport is less stereospecific in nature and allow the movement of charged ions. It also allows the movement of water molecule. The channel mediated transport is highly selective and not permanently open. The examples of the channel mediated transport are sodium, potassium, calcium, chlorine, etc. The channel mediated protein transport are of further three types. The first one is voltage gated ion channel, the second is ligand gated ion channel, and third is mechanogated ion channel. Let us discuss with the all one by one. The voltage gated ion channel. They are regulated by difference in the potential or electric charge across the plasma membrane. Let's see here. Let's see the diagram and try to understand the voltage gated ion channel. As you can see here, This is a deactivated, or this is a closed. This channel is a closed. And the charge on the plasma membrane, this is the extracellular space and this is the intercellular space. The charge on the extracellular space in the deactivated state is positive, and in the intercellular space it is negative. When the ion, this is the ion, when the ion bind to this channel, then the charge on the outer side of the cell become negative and the inner side of the cell become positive, that allow the entry of a charged ion. This is and this come inside the plasma membrane. And the next one, when the and this is inactivated or closed state. When this is inactivated or closed state, then this become totally same as the inactivated and deactivated state become totally same. And in each and in this the so this is voltage gated ion channel, and this channel is ion specific. The next one is ligand gated ion channel. These are not ion specific, as we discussed in a previous, that is voltage gated ion channel, that they are ion specific, but the ligand gated ion channels are not ion specific. They are regulated by the binding of the ligand. This is a ligand. These are regulated by the binding of the ligand to the target receptor, which is associated with the ion channel. This target receptor is associated with the ion channel. When this messenger, or we can say this ligand, bind to this target receptor, the ions it opens the ion channel. It opens the ion channel, and the ions move from outside to the inside in the plasma membrane. The example of the ligand-gated ion channels are acetylcholine dependent sodium ion channels on postsynaptic neurons and IP3 dependent intracellular calcium ion channels. The next one is mechanogated ion channel. In this ion channel, the conformational state depends upon the mechanical force applied. Let's understand with this diagram. This is the plasma membrane, this is the outer side and this is the inner side, and this is the mechanogated ion channel, and this is a closed at this time. This is a closed. When any mechanical force applied to the membrane, then this mechanical force converted into chemical or electric signal, and the mechanogated ion channels opens at that time. When any mechanical force applied to the plasma membrane, then these signals are converted into chemical or electric signals, and the channel is open, and the molecules can pass from outside to inside to the membrane. For example, the TMC in C. elegans and piezo in eukaryotic. Thank you.